the pharma biotech industry is the industry that really needs help. And I like to often quote Tom Insel in this context, where who was the former director of the National Institute of Mental Health and started MindStrong after spending a couple of years at Verily leading their translational neuroscience unit. He often says the house is on fire, and yes, we need to figure out the chemistry of the paint and really revolutionize the understanding and treatment of brain disease, mental illness. But we've got to put the fire out, right? I mean, there are tens of millions of people around the world that are suffering, either diagnosed or undiagnosed, probably hundreds of millions, suffering from mental illness and brain disease, and we don't have great drugs.
The way that I think about that, so if you're a pharma company, you're thinking about disease mechanisms and targets. Historically, you're thinking about things on like a genetic level, molecular level, protein level, cellular level, and yet the endpoints for Parkinson's, like a patient motor diary, or like some score of cognitive decline. And so there's just this massive gap between what you can test in a organoid or a petri dish or animal model and what your readout is in human behavior. And I think where like neurotech and devices are filling that gap basically in between. So they're saying obviously anything that you change in the brain at a cellular molecular level is going to be mediated, the behavior is going to be mediated by the neural circuit, right? It's by brain activity. And so having some understanding of what that looks like is, I think, pretty essential for having any real links between, clinical phenotypes and underlying disease mechanism. Otherwise, it's just a big black box, right? And that's hasn't served us very well.
Yeah, but you start seeing the connections, right? I mean, now there's papers about connecting within the central nervous system, the nigrostriatal pathway from the deep and from the cortical. The other day there was a paper that connected the vagal nervous with cortex. I mean, at the end, eventually, now there is the connectome project, right? The connectome is more central nervous system. Why not to extend it to the full body? That's going to happen as well.
And transforming an ecosystem, especially one that is as hard as the neuro ecosystem, neurotherapeutics ecosystem, is a long game. It's by no means, a short game. And what we're fundamentally, tackling is literally what Brian said, that animal models have failed us in the context of correcting human behavior. I mean, correcting mouse behavior is by no means indicative of whether human symptomology will be corrected. So I think what we really need in this space is a way to de-risk the clinical trial process by having better animal models that are much more indicative of how the human symptomology would be corrected or would progress. And we fundamentally believe, and we have seen a lot of these early proof points in some of the data sets that we have generated, our customers, colleagues, partners have generated, that the INBRAIN cell type specific circuit-based readout, even in a mouse model that is completely on a different planet in some ways from us as humans, in many diseases that do not rely on neocortex as a readout as an endpoint. Now, there's still a long way to go to really test that translational validity, but I can definitely say that in some areas we're already seeing, I will say, the early signs that the circuit-based readout can truly be much more predictive than state-of-the-art or even other animal-based readouts, behavior or non-behavior.